Welcome to the deep dive, everyone. Today we're going on a pretty fascinating journey into the intricate world of, well, your brain.
Our brains.
Yeah, our brains. We're diving deep way beyond just those familiar wrinkles and folds you see in pictures to explore the tiny universe within each brain cell.
Yeah, like the really, really small stuff.
Right. I mean, we're talking molecules here, right? You provided us with a mix of, well, some textbook excerpts and even some really cool scientific illustrations. So get ready for a pretty heady mix of like brain facts and visuals.
That's right. It's going to be molecular neurobiology time.
Oh, I'm ready.
And it's like going to be quite the trip.
All right, so lay it on me. What are we getting into today?
We're talking about a level of complexity that, well, it rivals the vastness of space.
Okay, hold on. You're comparing my brain to outer space.
Think about it like this. Astrophysicists study those fundamental particles, right? The building blocks of everything in the universe.
Okay, I follow.
Well, we're kind of doing something similar here. We're looking at the molecules, the proteins, the tiny interactions that make thoughts, emotions, literally everything that makes you you.
So instead of stars and galaxies, we've got neurons and synapses.
Precisely. And you might be surprised at how many of these tiny components even simple organisms have. Like the source material mentions fruit flies, those little guys buzzing around your fruit bowl.
Oh yeah, I know them.
Their tiny brains, believe it or not, have these things called mushroom bodies packed with 2,500 neurons, each connected to tons of others.
2,500 in a fruit fly brain. I mean, we have billions, right? That's a whole lot of connections to keep track of.
Oh, it is. And that's really just the beginning. Each neuron is like a bustling city, a metropolis of activity. DNA, RNA, proteins, all playing their parts.
I remember those from like way back in biology class. But give me a refresher. Why are these molecules so important for our brains to work properly?
Well, think of DNA as the master plan, the blueprint, the instruction manual for building and running your brain.
Got it. DNA is like the instruction manual.
Right. And then RNA, that's like the messenger. It carries those instructions from the DNA to the ribosomes.
Oh, right. Ribosomes. Those are like the little factories that make proteins, right?
Exactly. They take the instructions from RNA and use them to assemble proteins, the real workhorses of the cell.
Okay, so proteins are like the builders and maintainers of the brain.
More than that, they're messengers, communicators, gatekeepers, you name it. They do almost everything. And here's where things get really cool. The source material talks about something called exon shuffling.
Exon shuffling. Okay, now you've lost me.
It's basically evolution getting creative, taking these building blocks of genes and rearranging them, like a molecular Lego set.
Huh. So instead of making totally new genes, it's mixing and matching existing parts.
Exactly. And this process was super important in developing proteins crucial for brain function. For example, one of the sources talked about ion channels. Those are like gatekeepers that control how ions move across cell membranes.
Okay, I'm with you.
And some of these channels probably evolved through exon shuffling.
So by rearranging these building blocks, evolution created a whole bunch of different ion channels, each with its own, like, special properties.
Precisely. And that variety is essential for the brain's ability to make and transmit those electrical signals. But before we get into those signals, let's focus on these ion channels for a bit. How they work, what they do. The source material specifically mentions potassium channels. They're really important for regulating neuron activity.
All right, potassium channels. Why are they so important?
Well, potassium ions are always trying to escape from the neuron. Imagine them like kids in a classroom who can't wait to get out during a boring lecture.
Uh-huh, I get that.
And the potassium channels, they're the doors controlling their exit.
So controlling how much potassium can leave the neuron.
Yes. And there are all sorts of different doors, each with its own quirks and mechanisms. The source material groups them into three main types based on their structure: 2TM, 4TM, or 6TM.
Okay, I'm picturing doors, but honestly, I'm a bit lost on the 2TM, 4TM thing.
Imagine each channel is like a protein thread woven through a piece of fabric, which is the cell membrane. The number of times it crosses the membrane determines if it's 2TM, 4TM, or 6TM.
So the more times the thread crosses, the more complex the channel.
Exactly. And the structural diversity means there's a huge range of channel properties. They can respond to different signals and control neuronal activity in incredibly precise ways. What's even cooler is that by studying these channels in simpler organisms, like bacteria, we've learned a lot about the more complex ones in our brains.
Wow. So studying tiny bacteria can teach us about our own brains.
It really shows how connected life is, even at the molecular level. But now, let's talk about how these electrical signals, controlled by those ion channels, actually send messages between neurons.
Okay, so we have the basic building blocks and these channels controlling ion flow. But how does that turn into neurons actually talking to each other? The source material mentioned synapses and neurotransmitters. Where do those fit in?
Yeah, you got synapses and neurotransmitters. Those are the key players. Synapses are like points of contact between neurons, tiny bridges where messages get passed back and forth.
Okay, I get that analogy.
And the neurotransmitters, they're the actual messengers, the ones carrying the message across the bridge.
So it's not just like pure electricity going straight from one neuron to the next. There's this chemical component as well.
Exactly. It's a two-step process. First, you have that electrical signal, the action potential, traveling down the axon of the sending neuron, what we call the presynaptic neuron.
Right, the one sending the message.
Right. So it reaches the synapse, and that triggers the release of neurotransmitters from these little sacs called vesicles.
So it's like the signal runs to the end of a pier and tosses a message in a bottle into the water.
Uh-huh. That's a pretty good visual. And those neurotransmitter messages in a bottle, they float across this tiny gap, the synaptic cleft, and then bind to receptors on the receiving neuron, the postsynaptic neuron.
Okay, and what happens when the neurotransmitter finds that receptor?
That's when the message gets, like, decoded. It triggers a response in the postsynaptic neuron. It can either excite it, making it more likely to fire its own signal, or inhibit it, making it less likely.
So it's like a whole conversation going on between neurons, with some messages saying fire and others saying hold your fire.
Precisely. And this constant chatter, this back and forth of excitation and inhibition, is what shapes how the brain works as a whole. And the source material dives into this whole variety of neurotransmitters, each with its own like personality and role in the conversation. You've got glutamate, the most common excitatory neurotransmitter. It's involved in like learning and memory. Then you have GABA, the main inhibitory one. It helps keep things calm and balanced.
Well, it's like a balancing act between those two. What about other neurotransmitters? The source material mentioned serotonin and dopamine too, right?
Ah, those are the fun ones. Serotonin is often linked to, well, mood, sleep, and appetite. It's what gets boosted by antidepressants, for example. And dopamine, well, that's the reward and pleasure molecule, often implicated in addiction and motivation.
So each neurotransmitter has its own set of functions and affects the brain differently.
Exactly. And to make things even more interesting, each neurotransmitter can bind to multiple types of receptors, leading to different effects. The source material highlights two main types, ionotropic and metabotropic.
Okay, those are new terms. Need a little help here? What's the difference between those?
Think of ionotropic receptors like a simple on-off switch. When a neurotransmitter binds, it makes the receptor change shape, immediately opening a channel for ions to flow. This triggers a fast change in the neuron's electrical potential, either exciting or inhibiting it.
So it's a fast response, like flipping a light switch.
Right. Now, metabotropic receptors, those are a bit more subtle. They work through a chain of molecular events, often involving these things called G proteins and second messengers.
G proteins, second messengers. That's pretty complicated.
Think of them like relay runners passing a message along. The neurotransmitter binds to the metabotropic receptor, and that starts this chain reaction, eventually leading to changes in the neuron. But these changes happen slower, they last longer, and they can even affect not just the electrical stuff, but also gene expression and protein synthesis.
So metabotropic receptors are like setting a whole chain of events in motion, leading to more lasting changes in the neuron.
You got it. And this type of signaling is really important for things like learning and memory, where those long-term changes in how neurons connect are super important.
It's incredible how these tiny molecules, the neurotransmitters and receptors, have such a huge impact on everything we do, think, and feel.
It really is. And honestly, we're just scratching the surface here. There's still so much to uncover about this intricate dance of neurotransmitters, receptors, and signals. But for now, let's shift gears a bit. The source material takes us through this fascinating journey of how the brain develops and its amazing ability to change and adapt.
Brain development. That sounds pretty complex. I mean, how do billions of neurons know where to go and how to connect? It's like a massive construction project without any blueprints.
It really is a remarkable feat of self-organization, but it's not completely without blueprints. There isn't one single plan, but there are molecular cues and guiding principles that orchestrate the whole process. It starts with neurogenesis, the birth of new neurons, followed by cell migration, where those neurons travel to their final destinations.
Okay, so how do these neurons know where to go? It's like they have a GPS for brain cells.
You're not wrong. There are molecular signals, like signposts along the road, that guide migrating neurons to specific areas of the brain. They follow these cues, navigating this complex environment until they reach their designated spots.
So it's not just random, they're being directed based on molecular signals.
Exactly. And when they get there, they start forming synapses, those connections that let them talk to other neurons. But here's a twist. The source material talks about negative sculpting as a crucial part of this.
Negative sculpting, is that like brain cells being destroyed during development?
It does sound counterintuitive, but it's actually really important for refining the brain's wiring. Early on, way more neurons are made than are actually needed. Negative sculpting gets rid of the excess, pruning away connections that aren't being used or are not working right.
So it's like weeding a garden, getting rid of the weak plants to let the strong ones grow.
Exactly. And it's not just about getting rid of extra, it's about strengthening the connections that remain. The source material also mentions functional sculpting, where connections get stronger or weaker based on how much they're used.
So the things we experience, the stuff we learn, actually change how our brains are wired.
That's the power of neuroplasticity. It's how your brain adapts to new situations, learns new skills, and even recovers from injury. It means your brain is constantly being shaped by your experiences, even at the molecular level.
That's pretty mind-blowing. Just learning a language or playing an instrument can change the physical connections in our brains.
It really is. And this understanding has changed how we view the brain. It's not a fixed organ. It's dynamic, adaptable, constantly evolving throughout our lives.
So we've got this intricate network of neurons communicating through these electrical and chemical signals, and it's all being shaped by our experiences. But what happens when things go wrong? The source material gets a bit serious here and talks about neurodegenerative diseases.
That's where the beauty and complexity of molecular neurobiology meet the challenges of, well, human health. When these molecular mechanisms break down, the consequences can be pretty devastating.
The source material mentioned that even tiny mutations in genes coding for ion channels can lead to serious problems like channelopathies and some types of epilepsy. How can something so small have such a big impact?
Remember those ion channels we talked about? The ones that control ion flow across the membrane? Well, they're crucial for making and transmitting those electrical signals in the brain. And even a small change in a channel's structure caused by a mutation can mess up its function, leading to abnormal electrical activity.
So like a faulty switch, either stuck on, leading to too much excitation, or stuck off, blocking the signal.
Exactly. And this disruption can have a cascade of effects, leading to seizures, muscle weakness, all sorts of neurological symptoms, depending on which channels are affected and where they are in the brain.
Wow. And what about neurodegenerative diseases like Alzheimer's? The source material talked about amyloid plaques, tau tangles, and the APOE gene. Can you explain those?
Alzheimer's is particularly tough because it involves multiple things going wrong at the molecular level. It's like a perfect storm of cellular dysfunction.
Okay, let's start with amyloid plaques. What are they?
They're sticky clumps of protein that build up between neurons. Imagine gum stuck between gears, messing up communication.
So they disrupt the flow of information between neurons.
Exactly. And then you have tau tangles, which are twisted fibers of protein that form inside neurons. Think of them like tangles in a fishing line, preventing the neuron from working properly.
So one messes with communication between neurons, and the other messes up the neuron itself. What about the APOE gene?
That one comes in different forms, and some forms increase the risk of developing Alzheimer's and how fast it progresses. It's like a genetic predisposition that makes you more susceptible to those amyloid plaques and tau tangles.
So it's not just one thing. It's a combination of genes and environment that leads to brain cells dying.
That's what we understand so far, and why Alzheimer's research is so complex. Scientists are working hard to figure out the molecular details, hoping to find treatments and eventually a cure.
It's sad to think about these diseases taking away people's memories and abilities. But it's good to know that scientists are digging deep into the molecular causes, trying to find solutions.
Yeah, hope grounded in science. And that's what makes this deep dive so interesting. We're not just exploring the brain, we're exploring what makes us human. The molecules and interactions that create thoughts, emotions, everything.
This exploration has taken us deep into a single neuron, but the source material emphasizes that there's a whole other world out there, beyond the individual neuron, a world where cells interact, communicate, and form these complex networks.
Absolutely, and those interactions are key to understanding how the brain works as a whole. We got to talk about those connections, the points of contact, the molecules that make them happen.
It's like we're going even deeper down the rabbit hole.
We are, and what's cool is that it all comes back to those same principles. Membranes, proteins, the intricate dance of molecules. Let's zoom in on those cell membranes, the outer boundaries that define each cell's identity and how it interacts with others. The source material stresses how important they are.
Cell membranes. I vaguely remember those from biology class, but I'm guessing they're more complicated than I remember.
Oh, definitely. These membranes aren't just passive barriers, they're dynamic, constantly interacting with the environment and mediating communication between cells. The source material goes into their complex composition.
Okay, so remind me, what are cell membranes made of?
Think of it as a fluid mosaic, a base of lipids with proteins and carbohydrates sprinkled in.
Fluid mosaic. Can you maybe explain that a little better?
Imagine a sea of lipids, a double layer, with proteins floating within it like ships of all different shapes and sizes. Some proteins go all the way through the membrane, like channels or transporters, while others are stuck to one side, acting as receptors or signals.
So it's not a static wall, it's constantly changing and moving.
Exactly. And even the lipid base itself is pretty interesting. The source material dives into different types of lipids, like phospholipids, sphingosine, and glycolipids.
Okay, break down those lipid types for me. What are phospholipids and why are they important?
Phospholipids are the most common type in cell membranes. They have this cool structure: a head that loves water, hydrophilic, and a tail that hates water, hydrophobic.
So like a tiny magnet, one side attracted to water and the other repelling it.
That's a great way to put it. And because of this, phospholipids automatically form a bilayer, with the water-loving heads facing out towards the watery areas inside and outside the cell, and the water-hating tails facing in away from the water.
That's pretty clever. It's like they're forming a protective barrier to keep things separate. What about those other lipids, sphingosine and glycolipids? What do they do?
Sphingosine is another important one, especially abundant in the brain. It forms the backbone of sphingolipids, a special class of lipids involved in all sorts of cellular processes, like signal transduction and cell recognition.
So they're like specialized lipids with particular jobs to do.
Right. And glycolipids, those add a little sweetness to the mix. They're lipids with carbohydrates attached and are key for cell-to-cell interactions and communication.
So it's not just about building a wall, it's about creating a surface that can interact with what's around it.
You got it. And those interactions are crucial for everything from how neurons develop to how our immune system works. The source material even talks about the role of cholesterol in cell membranes.
Cholesterol, isn't that the stuff that's bad for our arteries? What's it doing in our brains?
Cholesterol gets a bad rap, but it's actually essential for a healthy brain. It helps regulate the fluidity of cell membranes, making them more stable and less leaky.
So it's like adding some stiffness to that fluid mosaic, right?
Exactly. And the right amount is crucial. Too little, and the membrane gets too fluid and unstable. Too much, and it gets rigid and inflexible.
The Goldilocks principle, again, everything in moderation. So we've got this lipid bilayer, this fluid mosaic that forms the base of the cell membrane. But what about those proteins we talked about earlier? What are they doing in this membrane world?
Those proteins are the real workhorses. They do a whole bunch of things, like transporting molecules across the membrane, receiving signals from other cells, and even catalyzing biochemical reactions.
So they're like specialized machinery, each with its own task, embedded in the lipid bilayer.
Exactly. And their placement is really important. Some span the whole membrane, forming channels or transporters, while others are anchored to one side, acting as receptors or signaling molecules.
And how do they get there? They don't just, like, magically appear, do they?
Definitely not. The whole process of making a membrane is super complex and fascinating. It's a carefully choreographed dance of cellular machinery, making sure the right proteins end up in the right spot at the right time.
Can you walk me through that process? I'm curious how the cell manages this whole construction project.
The source material uses a pretty cool example to illustrate it: the Xenopus rod outer segment disc.
Xenopus. That's like a type of frog, right?
That's right. And the rod cells in their eyes have these stacks of discs packed with visual pigment, the molecules that help us see.
So these discs are constantly being made and recycled, which makes them a good model for studying how membranes are made.
Exactly. It's like watching a cellular construction crew at work. The process starts in the endoplasmic reticulum, a network of membranes inside the cell where proteins destined for the membrane are synthesized.
So that's where the proteins are made.
Yeah. Then they're moved to the Golgi apparatus, another part of the cell that processes and packages proteins. Think of it like a sorting and distribution center.
So the proteins are made in one place, sorted and packaged in another. What happens next?
They get packaged into vesicles, little membrane-bound sacs that bud off from the Golgi. Imagine tiny delivery trucks carrying their cargo to the final
And
destination.
that destination is the cell membrane.
Exactly. These vesicles travel to the plasma membrane and merge with it, delivering those proteins right where they need to go. It's a super efficient and precise process.
It sounds like a well-oiled machine, each part making sure the right proteins get where they need to be.
It really is, and this process isn't limited to just the cell membrane. The source material also talks about axoplasmic flow, a process crucial for keeping neurons healthy and functioning.
Exoplasmic flow, that's a new one. What's that all about?
It's like a highway system inside the neuron, transporting stuff along the axon, the long, thin part of the neuron that carries electrical signals.
So like a supply chain, delivering goods.
Right, and it goes both ways. Stuff is transported from the cell body to the synapse, where neurons communicate, and back from the synapse to the cell body.
What kind of stuff are we talking about here?
Everything, from newly made proteins to organelles, the little structures inside the cell that do specific jobs. It's a constant flow of essential components, making sure the neuron can function and stay connected with other neurons.
It's incredible to imagine this whole transportation network inside the neuron, all happening on such a small scale.
It is, and this brings us to another fascinating piece of the puzzle: myelin. The source material emphasizes how important it is for making signal transmission faster.
Myelin, right? The fatty substance that wraps around axons. I've heard of it, but I'm not sure I understand what it does.
Think of myelin like insulation around an electrical wire. It prevents the signal from leaking out, so it travels much faster and more efficiently.
So it's like a supercharger for neural communication.
Precisely. It's crucial for the brain to work properly. Diseases that damage myelin, like multiple sclerosis, can have really serious effects on neurological health.
That's scary. What exactly is myelin made of, and how is it formed?
It's mostly lipids and proteins, kind of similar to the cell membrane itself. But the cool thing is that it's made by glial cells, these often overlooked cells in the nervous system.
Glial cells, those are the ones that support and nourish neurons, right?
That's them. And they play a huge role in making myelin. In the central nervous system, it's made by oligodendrocytes, while in the peripheral nervous system, it's Schwann cells.
So different types of glial cells make myelin in different parts of the nervous system.
Exactly. And how myelin is formed is pretty intricate. The glial cell wraps its membrane around the axon, squeezing out the cytoplasm and leaving behind these layers of tightly packed lipids and proteins.
So like wrapping a cable with layers of insulation.
That's a great analogy. And the structure of myelin isn't just random, it's very organized, with certain proteins playing key roles in its formation and stability. The source material mentions some genetic studies that have shed light on these proteins and how they work.
Genetic studies. So if there are mutations in those genes, they can cause problems with myelin.
Yeah, unfortunately. Mutations in genes that code for myelin proteins have been linked to various neurological disorders, which shows how important these proteins are for making and maintaining myelin.
It's amazing how something that seems as simple as insulation can have such a huge impact on how the brain works. But earlier you said there's a whole world beyond the individual neuron where cells interact and communicate. Can you tell me more about that?
Definitely. That's where cell junctions come in. They're specialized points of contact between cells that mediate communication and adhesion.
Cell junctions, are those like bridges connecting cells together?
That's a good way to picture it. And there are different types of cell junctions, each with its own structure and function. The source material mentions three main types: desmosomes, tight junctions, and gap junctions.
Okay, let's break those down. What are desmosomes?
Desmosomes are strong, adhesive junctions that hold cells together like rivets. They're important in tissues that experience a lot of mechanical stress, like skin and muscle.
So they're like the bolts holding a bridge together, providing structural support so cells don't get ripped apart.
Exactly. Then you have tight junctions. They create a seal between cells, preventing stuff from leaking through the spaces.
So like sealant preventing water from seeping through cracks.
Perfect analogy. They're crucial for maintaining tissue integrity and controlling what moves between cell layers. And in the brain, they're super important because they form the blood-brain barrier, which protects the brain from harmful substances in the blood.
The blood-brain barrier. So tight junctions are like gatekeepers, controlling what gets into and out of the brain.
Exactly. Then we have gap junctions, which are quite different. They're channels that directly connect the insides of two cells, allowing fast communication and exchange of small molecules.
So gap junctions are like tunnels between cells.
Yes, and they do all sorts of things in the brain like synchronizing neuron activity, facilitating metabolic cooperation, and even acting as electrical synapses.
Electrical synapses. So not all synapses use neurotransmitters.
Right. Most synapses use chemical neurotransmitters, but electrical synapses use gap junctions to directly transmit electrical currents between neurons. It's a much faster and more direct form of communication.
Wow, so gap junctions directly link neurons, letting them talk much quicker.
Exactly. And the source material mentions connexins as the proteins that form these gap junctions. These proteins come together to create channels that span the membranes of two cells.
So connexins are like the building blocks of those tunnels.
Precisely. And the interesting thing is connexins can open and close, controlling what goes through the gap junction. It adds another level of control to how cells communicate.
So gap junctions aren't just passive tunnels, they're actively regulating what passes between cells.
You got it. And they play many different but important roles in the brain. The source material mentions their involvement in how our retinas work, how the central nervous system develops, and even how neurons synchronize their firing.
That's a lot for these tiny channels to handle.
Yeah.
And the source material mentioned that diseases can affect gap junctions too.
Yeah, that's right. Mutations in connexin genes have been linked to all sorts of conditions like deafness, cataracts, and even Charcot-Marie tooth disease, a neurological disorder affecting the peripheral nerves.
So tiny channels, big impact on human health.
Definitely. It really highlights how important it is to understand these molecular mechanisms so we can develop treatments for these conditions.
This whole discussion of cell membranes and junctions has been really interesting. It's amazing to think of all these tiny processes working to keep our brains running.
And remember, this is just a fraction of what we know about them. There's so much more to discover about the world of cell membranes and junctions.
It makes you really appreciate just how complex the brain is and the incredible interactions happening all the time to make it work. But before we finish this deep dive, we have to talk about the heart of it all. The electrical signals that make the brain work.
Absolutely. We've talked about the building blocks, the communication networks, what can go wrong. Now let's get to the core of neuronal communication. The electrical signals that drive our thoughts, emotions, and actions.
Okay, electrical signals. This is where things get really interesting. The source material mentioned something called a membrane potential, which sounds important.
It is. It's like the basis of everything. Imagine a neuron as a tiny battery storing electrical energy.
Okay, a tiny battery. Got it.
And that energy comes from a difference in electrical charge across its membrane. That difference, the membrane potential, is created by ions, mostly sodium and potassium, being unevenly distributed inside and outside the cell.
So it's like having more positive charges on one side of the membrane and more negative charges on the other, creating this electrical tension.
Exactly. And just like a battery, this difference creates a voltage. In a resting neuron, this voltage, what we call the resting membrane potential, is usually around -70 millivolts, meaning the inside of the cell is more negative compared to the outside.
The source material mentioned an equation, the Nernst equation. Is that related to this? It sounds a little complicated.
We won't get lost in the math, but the Nernst equation helps us calculate the equilibrium potential for an ion. That's the membrane potential where there's no net flow of that ion across the membrane.
So it's like finding the balance point for each type of ion.
Exactly. And understanding this balance is key to understanding how different ions contribute to the overall membrane potential.
But the source material also mentioned that this equation only works for ions that can freely cross the membrane. And we know that cell membranes are picky about what gets through because of those channels and pumps, right?
That's a really important point. That selective permeability controlled by those channels and pumps is what allows the neuron to maintain that difference in charge, that electrical potential.
So it's like a constant tug of war between the ions trying to equalize and the membrane trying to maintain that separation of charge.
Exactly. And this back and forth, this push and pull, is what allows neurons to generate electrical signals, those bursts of activity that carry information throughout the brain.
Which brings us to the action potential. Can you break down how this signal is created and how it travels?
The action potential, that's like the language of the nervous system. It's a rapid change in membrane potential, a sudden burst of electrical activity that zips down the axon.
What triggers that sudden burst?
It starts with a little nudge. When a neuron receives enough excitatory input from other neurons, its membrane potential starts to become less negative. We call this depolarization. If this depolarization hits a certain threshold, usually around -55 millivolts, it triggers these voltage-gated sodium channels to open.
Voltage-gated sodium channels. Those sound familiar.
They're those protein gates we talked about,
Huh.
the ones that control the flow of sodium ions across the membrane. These specific ones are sensitive to changes in voltage. So when the membrane potential hits that threshold, they swing open, letting a bunch of sodium ions rush into the cell.
It's like a dam breaking, with a sudden flood of positive charge into the neuron.
That's a great analogy. And this influx of positive charge further depolarizes the membrane, creating a positive feedback loop that quickly pushes the membrane potential towards the sodium equilibrium potential.
So the neuron goes from being negatively charged to positively charged.
Just for a brief moment, yes. But it doesn't just keep getting more and more positive. The sodium channels shut off quickly after they open. And at the same time, voltage-gated potassium channels open up, letting potassium ions flow out of the cell.
So now positive charge is flowing out.
Exactly. And that outflow helps to repolarize the membrane, bringing it back down towards its resting potential.
Oh, it's like a carefully timed dance,
Mm,
with sodium rushing in, potassium rushing out, creating this quick spike in electrical activity.
you got it. And it's this dance, the precise timing of those channels opening and closing, that creates the characteristic shape of the action potential, that
What?
rapid rise and fall in membrane potential.
The source material mentioned that this action potential doesn't just sit there. It travels down the axon, carrying the signal to other neurons. How does that happen?
That's the amazing part. The depolarization caused by the action potential spreads to nearby parts of the membrane, triggering more sodium channels to open and pushing the signal down the axon.
Like a domino effect, one part of the axon triggering the next.
That's it. And it's this propagation, this wave of depolarization, that allows the signal to travel long distances, from the cell body where it starts all the way to the synapse, where it can be passed to another neuron.
And we talked about myelin before, that fatty insulation around some axons. Does that affect this signal propagation?
Myelin is super important for speeding things up. Remember, it acts like insulation, preventing the signal from leaking out. This lets the action potential jump between the gaps in the myelin, called nodes of Ranvier, making it travel much faster.
So myelin turns the domino effect into a high-speed relay race.
Exactly. And this speed is crucial for the fast communication that our brains need for complex functions. Imagine trying to do anything, play music, have a conversation. If those signals were moving super slow,
I can't imagine. The source material also mentions something called giant fiber systems in invertebrates as another way to achieve fast signaling. Are those similar to myelinated axons?
they achieve the same goal but use a different strategy. Invertebrates, like squid, have evolved these giant axons, much bigger in diameter than regular axons. This larger size reduces the resistance to electrical flow, so signals can travel much faster.
So it's like having a superhighway for electrical signals, less traffic congestion.
Precisely. It just goes to show how evolution has come up with all sorts of solutions to the problem of speed in how neurons communicate.
Wow, this has been quite a journey from the tiny world of molecules to these lightning-fast electrical signals zipping around in our brains. It's amazing to think that everything we do, feel, and think is ultimately powered by these tiny electrical pulses.
And remember, we've only just scratched the surface of this incredibly complex field. There's so much we still don't know about how these signals are generated, processed, and integrated to create all the amazing things our brains can do.
It really makes you appreciate the wonder of the brain and the intricate web of molecular and cellular events that make it all possible.
Definitely. The more we understand about the brain, the better equipped we'll be to solve its mysteries, tackle neurological diseases, and unlock its full potential.
Well, on that note, I think we've reached the end of our deep dive into the molecular world of the brain. I hope everyone listening has enjoyed this journey as much as we have.
Until next time, keep those synapses firing and those neurons connecting.